Zogopoulos, PhD student here at the BRFAA under the supervision of Professor Kanouliou from the School of Biology, University of Athens, and Dr. Leon Zogopoulos here at BRFAA. And today I'll be presenting the first some preliminary data from my PhD, and more specifically the human gene co-expression analysis, its second version, which is an RNA-seq-based web tool for gene co-expression analysis in Homo sapiens. Although this work was done as part of my PhD, it's actually, the end publication was actually a collaboration between many institutes, of course including the BRFAA and the University of Athens, and of course the University of Cyprus, the Aristotelian University in Thessaloniki, Ulster University from North Ireland, and all of this, and of course the School of Medicine in the University of Athens, and all of this was sponsored by HGCA. At the bottom I have the author list for our latest publication, and I have pulled out all the collaborators here from the BRFAA. So, to start, we should talk about gene co-expression. So, gene co-expression analysis combines mainly unrelated transcriptomic data that belong to the same organism and the same experimental platform. That platform can either be microarrays or RNA-seq, and it combines those samples, can be from different developmental stages, environmental conditions, and so on. In the case we select tissues that we select only a specific type of tissue, then we can say that the analysis is tissue dependent. But if we select many different unrelated tissues, we can say that the analysis is tissue independent. So let's say that we have two genes, the black and the red one, and in one sample, those two genes have a relatively low expression value. In another sample, which is unrelated to the other one, those two genes seem to get increased expression of the same rate. And we observe this kind of pattern across many different samples. So we can say that the relation between those two genes is that they are expressed with each other, since we observe that kind of expression pattern across all those multiple unrelated samples. And the thing is that about co-expressed genes is that they tend to participate in similar biological processes, and that is something that we're going to utilize in the process. So, we should talk about the workflow that we use for the construction of the HGCA website. And of course, the first part in everything is to get our primary data. For that, we selected the GTEx database, which stands for Genotype Tissue Expression, which is a very detailed consortia-type database, which contains multiple RNA-seq samples. We selected bulk RNA-seq in our case, and the thing is that they've taken all those samples under the same experimental conditions, and all of them are very detailed. We know exactly what everything came from, and you can see here at the right all the different types of tissues. We see that there's a very big variety of tissues. Everything is well represented, and especially we can see that samples from the brain are also very big variety. So to that end, we downloaded the gene read count data for around more than 16,000 samples containing gene count data from more than 55,000 genes. From 52 tissues, we decided to exclude cell lines since we wanted to study the global co-expression landscape under normal conditions in a human body. From around 950 healthy donors. The thing about GTEx is that they've made sure that at the time of death, those people were healthy, and that is the thing they want to utilize later on. So, the next part, actually, after taking our gene read count data, is to normalize them. For that, we selected the yarn, the Q-smooth normalization from the yarn R packets, and the good thing about QSmooth is that it normalizes samples under some assumptions. The first one is that the distribution of the samples at the same tissue should be the same, but that doesn't need to be the same across the entirety of samples. And that's a very good thing when we use databases and samples that are from multiple tissues, as in the case of GPEX. So we transformed our gene read count data to gene expression values, and we store them in a MySQL database, so it will be easier for us to use later on. The next part is the filtering of our samples. So we talked that there were about more than 16,000 samples. That is a good thing, that we get a lot of samples. But the problem is that this requires a lot of computer power. So, not only that, but it's also the case that we want to reduce the tissue bias from all the different samples. For that, we want to do some kind of selection on them, and in order to do that, we decided to do it in the following way. First, we got the expression values belonging to those samples, and for each sample, using the Pearson correlation coefficient, we calculated the pairwise correlation between each sample, and the Pearson correlation coefficient gives us a value of -1 to 1, where -1 shows an anti-correlation between two samples, while 1 shows us that those two samples are almost equal. And we calculated that value across the entirety of the pairs of all samples, and in order to transform them from negative values to non-negative values, we performed this very simple transformation to transform it to distance values. From that, we resulted in a sample distance matrix, which shows us the distance between each of the samples, and we used that as input to the UPGMA clustering algorithm. You may know that as the average lineage algorithm, and that resulted in a sample tree which resembles phylogenetic trees, and on that tree, each leaf was represented by one of our samples. And now, in order to perform our sample filtering, we used our own auto-pruning algorithm that would clip leaves that were adjacent to each other and thus were very close to each other. And from more than 16,000 samples, we decided to keep only 3,500. And let's zoom in on that for a bit, and you can see how the tissues, the samples and the respective tissue were originally clustered in using UPGMA, and you can see that all samples of the same tissue are really clustered very well together. It's also a good indication about the high quality of the GTEx data and our normalization process. And now, as far as the filtering is concerned, the long lines represent samples that were kept, and the short lines represent samples that were eventually pruned. So, after we have our representative 3,500 samples, it's time to perform the actual gene co-expression analysis. This step isn't that much different from what we described when we performed the pairwise correlations between each sample. So now the only difference is that we have the expression values corresponding only to the 3,500 samples that were kept. Now, we also do Pearson correlation coefficient, but this time only between the genes. So we compare the expression values of the gene in all samples versus another gene in all samples, and we can get another Pearson correlation value for each pair of genes, which we also use to transform to a distance value. Thus, we result in distance matrix between each of the genes, and this is also used as input to the UPGMA algorithm, resulting in the final output of HGCA, which is actually a gene co-expression tree in unique format, whose leaves correspond to all our studied genes, which are more than 55,000. And on that tree, if we zoom in on a specific branch, we see a gene of our selection and its co-expressed partners, so genes that are close to each other in adjacent leaves, say, that constitutes our Coexpression results. So moving on. The last part in performing Coexpression analysis is our result evaluation. In order to do that, we use enrichment analysis of biological terms, and we do that on the specific clade that we have selected that contains our supposed co-expressed partners. The enrichment analysis is performed using the hypergeometric distribution for each specific term that we want to test, and this results on p-values that are adjusted for false discovery rate. So a p-value actually shows us the possibility that the term that we found as overrepresented has actually happened by chance. So the lowest the p-value, the better confidence we get that this term truly is truly enriched for our co-expressed genes of interest. So in HGCA2 we offer many types of enrichments from multiple biological terms, starting from all three aspects of gene ontology. We have gene pathways and metabolic pathways from KP and wiki pathways. We have protein families from PFAM. We have gene disease associations from OMI and GenNets. And finally, we have experimentally confirmed transcription factor targets of genes from REMAP and ENCODE. So here you can see the latest version of the HGCA website. It's available here in our local website, in our own website. And it's also available in the Elixir BioTools under this link. And on the main page, the only thing that someone sees as an input is this field right here, which is used to insert a gene name. If someone is not aware of a certain gene or what they would like to test, they can either use some of our examples right here, or they can search gene cards to find the actual name of the gene they want to test. So in this case, let's say that we want to test for a ribosomal protein, RPL24. So we input its HGC symbol, and the website auto-completes depending on the available genes on the database. So we write that one, we choose it, and we press submit. And the only thing that we see is the results page. So let's go through it a bit because there are a lot of stuff here, and I might want to explain them. So the thing we first get is a co-expression clade that contains our input gene highlighted in yellow. We call it the driver gene from on forth. And on that co-expression clade, we see genes that are co-expressed with our gene of interest, with our input gene. The user can increase or decrease the size of that clade by adding or subtracting internal nodes. Internal nodes are the connecting nodes starting from our driver gene up to the root of the tree. And in addition, the user can also export or download the tree in unit format, the list of the co-expressed genes, and also not only locally, but also to many other secondary websites performing enrichment analysis or other kind of analysis, such as gProfiler, EnrichR, Flame, GeneMania, Pathway Commons, and Stream. Finally, we mentioned that HGCA2 includes their built-in enrichment analysis categories. I have mentioned them here, but you can also, I mentioned them earlier, but you can also see them right here. And by selecting each one, you get this table at the bottom, which includes the top enriched terms for this specific gene co-expression clade. So, since we've mentioned how the website was created and how it works, it's time to move on to some of the results and some of the use cases we tested on HGCA2. So to do that, first we select a driver gene of interest, we input it to HGCA2, and we perform a manual exploration of the default Coexpression output. To do that, we increase or decrease the clade. In order to find the optimal gene plates, we have many indications for that, including gene partners that we know exist and enriched terms. And then, from the gene partners of the tree that we select and the enriched terms from all kinds of analysis, we try to interpret the results through asking experienced partners in that field and through bibliographical search. So, let's start. The first case was actually STAT1, transcription factor, and we know that STAT1 is induced by interference, and it also plays a very well-documented role in regulating the other genes related to different response to viruses and various stimuli. And on that tree, we see STAT1 and many other co-expressed genes that are also induced by interference, such as the OAS family of genes and the IFI family of genes. Naturally, most of the terms that were found as enriched on that specific tree are related to different response to viruses with very low p-values. And we also see many terms that are related to viral infections right here. Finally, one other interesting thing is that STAT2, of the same family of STAT1, was found to be regulating more than 2/3 of the genes of the subtree, and we might say that plays a role in the co-expression of all those genes. Then we try to test the nature histocompatibility complex class 2 genes, and for that we used HLA-DNA as gene of input. Initially, HGCA2 resulted in the subtree that is highlighted, that is in the green border right here. And the enriched terms were, of course, related to antigen crossing representation by MHC class 2, and most of the terms were also related to that specific function. Now, at this point, we decided to also expand the co-expression clade, resulting in a much larger clade that is included in the red border. By performing a recent analysis on that greatest clade, we got many more general terms that were nonetheless related to adaptive immunity. Another case for HLA genes is NLRC5, which has another well-documented role in regulating MHC class 1 genes. So by using NLRC5 as input to HGCA2, we naturally got genes of MHC class 1 family, and we also got the enriched terms to support this finding. Another interesting case is that of the growth hormone family. The CSLH1 belongs to that family of growth hormones, and its expression is mostly in the placenta. In human, growth hormone is actually comprised, is actually comprised of two genes, growth hormone 1 from the actual growth hormone and GH2 for the growth hormone during placental growth. And the thing, after we use the CSH1 as input, the first thing that we notice is that it is mostly co-expressed with the genes of the growth hormone family that are also expressed in placenta, while GH1 and prolactin are on a different rate and on a different plane, yet they are really close to each other, and they also belong in the same co-expression plane. Naturally, the terms that we got as enriched are related to the response to growth hormone receptor signaling and regulation of growth. Furthermore, many other diseases and pathways that we got are diseases that are related to growth. Finally, by performing chromosome band analysis, we found that most of those genes are actually enriched for the chromosome 17 genetic locus that contains the growth hormone genes. Another case was the metallothionein family, and we used the MT1N gene for that purpose. HGCA2 resulted in a clade that contained many metallothionein family genes. Some of them were pseudogenes, some of them were actual genes, and most of the enriched terms were related to the metallothionein function, which includes time response to copper ion, detoxification of copper ion, and heavy metal homeostasis. To be with the current times of COVID-19, we also decided to test the TMPRSS2 gene, which codes for transmembrane protein. And we know it from current stuff that TMPRSS2 serves as has a general use for epithelial homeostasis, but many viruses, including COVID-19, use TMPRSS2 for invasion. And we got a large subclade, and the enriched terms from that subclade were actually related to virus attachment to epithelial cells by getting terms such as cell-cell junction assembly, epithelial cell differentiation, and cell adhesion molecules. Another really interesting case is that of this gene right here, which is an ORF, but it's actually known with another name, XP32, also known as spin-specific protein 32. We used that gene as input to HGCA, and HGCA resulted in a Coexpression clade that included mostly LC genes or late conified envelope genes. The late conified envelope genes are responsible for keratinization and are mostly expressed in skin. And naturally, we got such terms as being enriched. Epidermis development, keratinization, and vitamin D receptor pathway. And chromosome band analysis showed that all of those genes are located in a specific part of chromosome 1, which is also known as the epidermal differentiation complex. And as we can see right here, this is an image from the Ensemble genome browser. Here is the gene that we tested as input, and we can see that all the other late conified envelope genes are around that same genomic location. So we can say that it is highly possible that the co-expression of this specific gene is correlated to the localization, the decolocalization of all those genes at this point. Then, the last case that I want to present in detail is that of neuropilin-1, which is a gene that it serves as a receptor for VEGF, and it's also a gene with functional immunogenesis. And naturally, the resulting co-expression during co-expressed genes resulted in terms that are related to the specific functions of the genes, such as vascular development, cardiovascular system development, so on and so forth, and of course, pathways that are related to its function as a receptor for VEGF. Some other examples that we tested in HGCA2 include the glucocorticoid receptor, where we found co-expressed genes that relate to its function. We also tested for heat shock proteins, both stress-induced, those two, HSP90AA and its parallel. We found that they are also in the same co-expression clade, and they are co-expressed with genes that include functions related to their activity as chaperones. And also, we tested the HSP90B1 heat shock protein, which seems to be constitutively expressed, and we found genes that relate to its functions as well, but are not stress-induced. Another interesting case is that of the olfactory receptor genes. So olfactory receptors constitute a family of genes responsible for the sense of smell that it would like 800, 900 members, but around 60% of those are actually pseudogenes. So, in a specific case where we tested for factor receptor genes, we found 400 gene clade that contained 220 of factor receptors, with 40 of them being pseudogenes. We saw that all of them were grouped together, yet that was kind of strange to us, because it makes sense, but we did not have a way to actually prove it, so to actually understand that. Because we know that the factor receptors actually follow a monogenic expression. So we tested the actual correlation values of those genes between, and we found that they were all close to 0. Yes. So those genes were grouped together, but there was no actual correlation to support that. So what we thought is that they had some slight correlation between them, but they were also not correlated with any other gene, so the UVGene algorithms actually grouped them all together. Also, we tried to test the case for 2 neuromuscular diseases, that of ALS and LGMDs, and we want to thank our collaborators from the University of Ulster for helping with this analysis. And they provide us with lists from Orphanet and from their own experience in bibliography. And we tested many ALS causal genes, and on all those cases we found that although center single genes were co-expressed with other genes with related functions, there weren't really many genes, there weren't any genes actually that were located on the same subclade with each other. On the other hand, for the LGMD causal genes, we not only found genes that were in clades with related and reached functions, but we also found many of them being located, inhabiting the same co-expressed clades. Finally, we also tested for antigen genes, since we found out that through experience using the tool, that many of the genes tended to be co-expressed with their antisense. This can be either due to the same sequence in the sense of antisense genes, but there could also be a regulatory role of the antisense genes to their sense genes. Finally, since we can't really judge the quality of expression to a vacuum, we decided to compare HGCA results with 4 other co-expression tools. 5 if you count the gene friends, actually has 2 versions. So in order to be as just as possible, we got a list of around 25 genes per tool per driver gene case, and we used a string database, webtool. String is actually a website that creates protein-protein interaction networks depending on a given list of genes. And by using that, we wanted to have a very unbiased opinion about the co-expression results. So to do that, we did not select any co-expression relationship. We only want to select all the other cases, relationships that string includes. We included a high confidence cutoff, and we used the following metrics, which not only include network metrics, but also biological process and chain enrichments. So this is one example out of all the cases that we tested, and we can see that HGCA2 and Co-Express-TB produce dense networks, followed by gene friends and RHS4, and the SRA version of gene friends and SIG are next. So by ranking those websites not only from their network metrics, but also from the enriched terms that they produced, we resulted on the same conclusion. So apart from the test of string, we also want to say that the results of each co-expression webtool really vary on a case-by-case scenario for each different gene. And furthermore, there are many webtools that do not include built-in enrichment analysis, which is something that HGCA2 does. And finally, the thing that one co-expression webtool has to take into account is that the result presentation has to be comprehensive by the users. So we have co-expressed GMVs, co-expressed gene co-expression networks, and gene co-expression traits. And although a list can sometimes be useful, a network can get really weird to explain sometimes, but co-expression traits is something that is really understood by biologists. It also coincides with phylogeny. And if we want to get a discussion out of the results, we can say that in general, co-expression analysis really depends on the type of samples that is used and on sample reprocessing and batch correction. I mentioned that since there are not only HGCA but also gene friends use GTEx as a main source for their data, yet we got really different examples, really different results out of them. That means that it seems that maybe HGCA2 normalization method that we use could be superior to that of gene friends. And finally, the updated biological terms need to be used so we can get the latest results for gene annotation. Finally, it seems that RNA-seq data should be referred to microarray at this point, not only due to the high amount of data that are produced, but also because RNA-seq are much more sensitive and allow for the study of all transcripts of an organism. Also, high-quality data from consortia such as GTEx that we use here in this case seem to produce better co-expression results. That's it, not only because we do not have to consider batch correction since it all comes from the same study, but also it seems to the case that we tested for gene friends in GTEx and gene friends in SRA version containing multiple samples from different studies, the gene friends GTEx version performed better. And finally, as food for thought, single-cell RNA-seq data at this point might be able to highlight the intricacies of gene expression, as in the case of factor receptors. And thus, that could result in a more clear gene co-expression analysis, which means that we might also get higher correlations between genes. So, what is the take-home message today? HGCA2 is a conditionally dependent RNA-seq-based co-expression analysis webtool that we showed to be able to reproduce known biology in a variety of gene use cases. So, that gives us the confidence to say that HGCA2 can produce experimentally verifiable hypotheses for the users. And that means that HGCA2 is able to discover potential and functional gene partners and also genes that follow the same pathway through a guild by association approach. We try to make HGCA2 tailor-made for molecular biologists through its simple design and easily interpretable results. But we also cater to programmers, since we include an application program interface, so you can easily programmatically get the results. There are two publications that are related to this work. The first one is our recent one says that actually describes the tool itself, and a previous publication is a review article about co-expression analysis, and we recommend everyone to watch that, to read that as a primer for gene co-expression analysis. At this point, I would like to thank everyone who worked with us with this project, our supervisors, Professor Vasileios for her supervision not only during my master thesis, but also during the PhD right now, and of course Dr. Vasileios for all his help throughout all these years and his constant help support. For the development of the webtool, I want to thank Dr. Apostolos Malatras and Konstantinos Kyriakidis and Chris Vasileiou for doing the initial analysis on GTEx data. For the use cases validation, I'd like to thank all the partners here at the BRFAA, Chryssanthi Charalampous, of course, Dr. Marios Zogopoulos and his PhD students, Marianne and Magdalena. I'm really glad you're here to watch me today. And from the School of Medicine, I want to thank Professor Chrysos and Eleanor Hiliani. And finally, our partners from the Also University, Dr. Dugues and Professor Dudi. And of course, every one of you who watched this talk with me today. Thank you very much.
Thank you, Vasileios, for this excellent talk. So, questions? Okay, I will start. As you know, the new trend in science, in immunology, and other fields, is the discovery of new cell types, of unknown cell types. You mentioned the single cell analysis, that it is a very advanced way to approach an expression patterns of unknown cell types. Do you think that such tools as HGCA can be applicable this type of discoveries?
In this case, I do not think that gene co-expression can really help in discovering new cell types. However, there are many new methods that use single cell data and perform all those kind of artificial intelligence, machine learning type algorithms that are able to result in much better kinds of correlations between genes. If we want to talk about cell type discovery, that's an entirely different thing, and that should mostly be done, requires a different kind of procedure. Thank you.
Thank you for this very interesting presentation. I have two questions, but first of all, I have to declare that I'm quite ignorant in this field, so they may be quite naive and may be difficult to answer. The first one is, I noticed that you cataloged several genes that were associated, and I believe all of them were coding for proteins.
Yes.
In your method, were they discovered any longer coding RNAs that would have associated with a different co-expression?
Okay. Yeah, we did manage to. There are many cases you can see on the clades themselves that there are indeed included some long-encoded RNAs inside the co-expression clades. Not only that, but in the antisense gene case, we also found many long-encoded RNAs that seem to also perform a regulatory load on their sense genes.
Okay, thank you. The second question I have is the following. By co-expression, I guess you mean that genes that are expressed differentially or specifically at higher level. In other words, I'm asking, would your method uncover genes that are down-regulated at the same time?
Yes, gene co-expression analysis is actually a very systems biology approach. So in this case, if we see two genes that are down-regulated at the same time, we can say that they are co-expressing each other, but we have to observe this pattern in many different samples to be able to say that with confidence.
In addition, so many different samples for your approach does not mean many different data sets.
Many different? Excuse me?
Data sets. What's the difference? You have many single cells, you have many different data sets.
Don't move your hand.
You have many different cells, you have genes, you have many different data sets, depending of the origin. Or you have many different tissues. What is the difference for your two?
In this case, we wanted to include as many different tissues as possible, but on the condition that not one tissue overshadows the other tissues. So that's why we performed our sample filtering, so we could get an equal representation of them. The more tissue types that we get, the better it is for us to get the whole co-expression landscape of the organism in question. Now, to move that a little bit further, at the end, we recommend that, if possible, all samples should be from the same study. That's why we recommend GTX or maybe other consortia for this purpose, since it is very difficult to perform batch correction when you get samples from many different kinds of studies. And that could actually impact the results of expression.
Thank you. Another question? No questions. Okay, thank you, Vasileios.
Sorry.
I would also like to ask something. Yeah.
Of course.
Congratulations, Vasily, for the very nice presentation and the excellent work, and also to the whole team for the collaboration with the whole team, especially with Dr. Yannis Michalopoulos. I also like to, I think it would be nice to make a comment on the most crucial part of the work and the most difficult part for you of the work. 2 comments. So what was the most crucial part of the whole?
You mean
yeah.
for me to comment on that?
The most crucial and the most difficult for you, especially.
Okay, of course, I think that the most crucial part was actually getting that very big data set of 55,000 genes and 3,500 samples to be able to be constructed. That we tried to find many ways to do that. It actually took us a lot of time to actually get the final result, but simply by the time we got that, we were able to move forward very easily. And the next thing, the most difficult thing for me, was actually the result interpretation. Of course, I can, at this point, I think I'm able to see if there are links between some of the genes in the co-expression results of our tree, but it is not always very easy to dive deep into the results and deep into the molecular mechanisms that actually gives us this co-expression. And that's why I'm very grateful to all the collaborators for helping us with this.
Thank you very much. Thank you. Congratulations again.
Another question? Okay. Thank you so much, Vasily.
One final note, if anyone has any questions about the webtool or they would like to do a presentation, a live presentation of it, they could always ask me or they can visit our website. Thank you very much.